Welcome everyone, I am Shaptapurni from BioLive. Hope all of you are doing very well. So today I am going to discuss with you some short questions from Colorimetry chapter. And this is very important for those students who are undergoing zoology honors for their practical viva examination, and also for those who are undergoing the MSc in zoology. So let's begin. The first question asks, what do you mean by colorimetry and colorimeter, and what is the principle behind the colorimetry by Lowry's method? So see, colorimetry is a scientific method, whereas colorimeter is an instrument used for that. Okay, so let's see what is it. Colorimetry is a scientific technique used to determine the concentration of a substance in a colored solution by measuring the absorption of monochromatic light of a particular wavelength by the colored solution following Beer-Lambert's law. Okay, so as the name suggests, colorimetry or colorimeter is related to some kind of colored solution, and you need to know the concentration of the substance which is dissolved in that colored solution. Okay, so how will you know? For that, you need to measure the absorption of a monochromatic light of a particular wavelength by that colored solution. Okay, following the Beer-Lambert's law, then you can only get the required concentration of the unknown sample solution. And for that, we use the colorimeter. So colorimeter is the instrument which is used to determine the concentration of any substance in the colored solution following Beer-Lambert's law. Now, what is the principle behind the colorimetry by Lowry's method? So, see, in colorimetric estimation of protein by Lowry's method, there are two reactions. First, protein reacts with alkaline copper reagent and forms coordination complex, also known as protein copper complex. Okay, how do they form? Due to the reduction of Cu2 plus ion to Cu plus ion with the lone pair of electrons of nitrogen atom of the peptide bond, as in biuret test. Okay, then these protein copper complex or coordination complex reacts with phosphomolybdotungstate of the phenol thiocalcium-phenol reagent and forms a bluish compound named heteropolymolybdenum blue by the tyrosine and tryptophan amino acid residues of the protein. So first, your protein reacts with the alkaline copper reagent. Okay, how do they react? The lone pair of electrons of the nitrogen atom of the CONH bond, that means the peptide bond, reacts with the Cu2 plus ion of copper reagent. Okay, and they form a complex known as protein copper complex or coordination complex. Then these coordination complex again reacts with the Folin thiocalcium-phenol reagent. Okay, what is that? The name is phosphomolybdotungstate of Folin thiocalcium-phenol reagent, and they then form heteropolymolybdenum blue, a bluish compound. How do they form? In this reaction, actually the tyrosine and tryptophan amino acid of your protein take part. Okay, and thus a bluish compound is formed. Now you need to find out the absorbance of that bluish solution. Okay, by the Beer-Lambert's law, as we know, and then you can find out the concentration of your unknown sample. So then the protein present in the bluish compound solution absorbs monochromatic light of a particular wavelength in colorimeter following Beer-Lambert's law, and by plotting the OD value of the unknown protein sample against the standard curve prepared using known concentration of protein sample, we can determine the concentration of the unknown protein. Okay, so this is the principle behind our colorimetric estimation of protein by Lowry's method. Now, who proposed the Lowry's method? It was proposed by famous biochemist Oliver H. Lowry in 1951. Next, what are the functions of sodium carbonate, Rochelli salt and copper sulfate in alkaline copper reagents? See, sodium carbonate actually makes the environment alkaline. Okay, whereas Rochelli salt, which is also known as sodium potassium tartarate, the tartarate part of this Rochelli salt actually acts as a chelating agent. It chelates the lone pair of electrons of nitrogen atom of the peptide bond with copper iron and stabilizes the protein copper or coordination complex. Whereas the copper sulfate actually provides the copper for chelation reaction, okay, with the protein as in biorate test. Now coming to in which ratio the sodium carbonate, Rochelli salt and copper sulfate is present in alkaline copper reagent. The ratio is 98:1:1. Next, what are the components of falling cycaltude reagent? The components are sodium tungstate, sodium molybdenate and phosphoric acid. Now, what is Beer's law? So, simply remember Beer's law states absorbance is directly proportional to the concentration of your solution, colored solution. So, when a monochromatic light passes through a homogeneous colored solution, the amount of light absorbed by the solution is directly proportional to the concentration of the solution. A varies as C. Now, what is Lambert's law? Lambert's law states that when a monochromatic light passes through a homogeneous colored solution, the amount of light absorbed, that means the absorption of the light by the solution is directly proportional to the length of the absorbing medium, that is A varies as L, where L is the length of the absorbing medium. What is the length? That means the length, the thickness of the cuvette, okay, the cuvette you are using for colorimetric estimation, the thickness of that is your length. So A varies as L. So going to the next question, define absorbance and transmittance. See, if this is a substance whose length is small l, and this is the intensity of the falling light or incident light known as I naught, and this is the so obviously some light will be absorbed and after that rest will be transmitted, and the intensity of the transmitted light is I. Okay, so absorbance means absorbance, which is also known as the optical density or OD, is the quantity of light absorbed by the colored solution. So, how much amount of light is being absorbed by your colored solution is the absorbance or optical density. It is actually a log intensity ratio of falling light upon the substance to the light transmitted through the substance. That is why we can also write OD or A equal to log 10 base intensity of the falling light, that is I naught, divided by intensity of the transmitted light, that is I. Okay, we can also write it as minus log 10 base I by I naught, just flipping these two. So this thing I by I naught is actually T, so it is also minus log 10 base T. Okay, so now coming to the what is transmittance. So obviously transmittance refers to the amount of light that passes through a colored solution without being absorbed. So transmittance can be represented as the log ratio of intensity of transmitted light by intensity of falling light, just the opposite of A. Okay, now coming to what is BSA and from where is it obtained. So in colorimetric estimation of protein by Lowry's method, we generally use BSA. BSA stands for bovine serum albumin, okay, which is the fraction, also known as fraction 5, a protein derived from the bovine blood plasma. Okay, so it is derived from blood plasma of cow. It has 583 amino acid residues bound in a single chain, cross-linked by 17 cysteine residues. Among these 17 cysteine residues, 16 cysteine residues form the 8 disulfide bond, and one is represented as free thiol group. Okay, it is widely used in analytical chemistry due to its easy availability in the market. Okay, got it. Now coming to the next question. Why hand incubation is needed in Lowry's method? See, in Lowry's method for two reactions, that is the first reaction with copper reagent and second reaction with falling cycaltude reagent, the required temperature is 37 degrees Celsius, which is our body temperature. So for that reason, the reaction is performed by hand incubation. Okay. Next, the OD value is measured in colorimeter at which wavelength and what filter is used for that. See, OD value is measured in colorimeter at an around 650 nanometer wavelength using red filter. Now, what is the function of this filter in colorimeter? The filter in colorimeter actually converts polychromatic light into a monochromatic light of specific wavelength. Next question. In photoelectric cell of colorimeter, light energy is converted into which energy? It is converted into electrical energy. Next question, what is standard curve? Okay, so see, standard curve in a model graph, it is also known as calibration curve. Okay, it is used to determine the concentration of unknown substance in a sample solution, comparing to a set of standard samples of known concentration. When someone is giving you a sample with unknown concentration, then obviously you will get the OD value from the colorimeter, but only getting the OD value does not give you the concentration of the protein of the unknown sample. Okay, so for that you need to plot your OD value against a standard curve which is prepared using some standard samples with known concentration. Okay, so obviously just measuring and thereby obtaining the OD value does not give protein concentration of the unknown sample. So to obtain that, a model graph is plotted using the standard curve, keeping protein concentration on the x-axis and OD values on the y-axis. Okay. Now coming to the next question. Why can't we measure concentration of albumin, egg albumin, by colorimetry? See, egg albumin does not contain tyrosine and tryptophan, which are needed for the reaction with Folin cyclotube reagent. So obviously we cannot measure the concentration of egg albumin in colorimetry by colorimetric measurement. Okay. Now very important question. Does the OD value depend on the cuvette's thickness? What do you think? Yes, obviously it depends, because according to Lambert's law we know that A varies as L. Okay, so obviously if the L varies, see here this is one cuvette and this is another cuvette. Concentration is similar. Okay, so here, but here the length of the cuvette is smaller than this, so L2 is greater than L1, so obviously absorbance by the L2, that is this cuvette, A2 will be greater than A1. Okay, so absorbance will obviously depend upon the cuvette's thickness, or OD value obviously depends. Now coming to the last question, what is a blank and what blank is used in colorimetric estimation and why? See, blank is a solution that is used to calibrate the colorimeter or adjust the colorimeter to zero absorbance. Generally we use distilled water as blank solution. Why? Because generally we dissolve the BSA, the protein sample, in distilled water. So distilled water is the solvent. Now obviously maybe it is transparent, but obviously there are some impurities. So when you are measuring the sample solution, obviously they will also encounter the absorbance by its solvent, that is the impurity of the distilled water. So to negate that, first of all we need to set only the distilled water as blank and its absorbance as 0. So generally distilled water is used as blank because it is the solvent where protein is dissolved. To make the colorimeter ignore any light absorbed by the solvent, that is the distilled water, and only to measure the light absorbed by the chromophore in the colored solution, blank is used to adjust the machine at 0 absorbance. I think you have got it. So if you have any more questions on this chapter, please do comment in the comment section below. And if you like this video, don't forget to like, share and subscribe. Thanks for watching and happy learning.